those microscopy pictures of tumor cells. They look really nice, but actually they are not. So we know that cancer actually is like a term to describe broad disease. That is like a gathering of cells with the ability to proliferate uncontrollably. And what is basic now in the cancer research, researchers who are working on cancer, they are most of the time working on cancer cell lines. So cancer cell lines are immortalized cells which are isolated from patient samples, and they are able to proliferate in the laboratory, in the flask, which of course the researchers need to provide a certain condition that they are alive, and we will learn about that today. So researchers for many years, because this cancer is known since very long time, but researchers for many years tried to figure out how is it possible to actually try to find drugs for cancer and work with that, because of course the basic thing is to try to isolate some cells which are able to survive, and then try different drugs and different tests how to overcome this disease. So this was possible in 1951 in USA, when one of the researchers in Johns Hopkins Hospital had a patient called Henrietta Lacks. She had a cancer, cervical cancer, and he isolated cells from her tumor after cutting it, and it was the first cells that researchers were able to cultivate in a laboratory. And after they were trying to figure out actually what is the reason why those cells survive while many years of research didn't bring anything, because they tried to make those cells being alive in the laboratory, but it never worked. And they figured out that those cells has active version of telomerase, which is enzyme which we have telomeres, which when they are shortening, this is why actually we are aging and dying. So the cells where it doesn't have any more telomeres, it's dying. So when this enzyme telomerase is active, it's possible that the cells still can proliferate. And this was the hallmark of these cells. And this became also afterwards a genetic manipulation of different cells, which researchers isolate with different patient tumors. This was the mechanism how they managed to immortalize cells. Nowadays there is really many, as you can see listed here. I even don't know many of them. It's just a list, just to show you that nowadays there is a lot of different mechanisms how researchers are manipulating isolated cells from cancer to be able to cultivate them in the laboratory, and to be able to prevent that they die at some point. So all cancer cells which are now in use in the laboratories, they are immortalized. You can cultivate them until the end of your life. So that was just a short introduction, just a few words. Now we come to more important part, which is how to handle cells, cancer cells. So the most important thing is that everything what you do is in sterile condition. So you cannot open, because cells are cultivated in the flask, you cannot open them outside. You need to open them under the hood, which is designed to enable a sterile condition for this kind of works. And everything what you use for cancer, like medium, which is their food, or PBS, or whatever other solution, they all need to be sterile, so they all need to be all autoclaved. Every plastic or glass material, pipettes or tubes, whatever else, everything needs to be sterile. So everything what is prepared now for the practical course, everything is already sterilized. The basic condition of cultivating cancer cells is first of all the place where they stay all the time. So it's an incubator, a closed incubator where it needs to be 37 degrees, and also there need to be provided CO2, which enable to maintain physiological pH. And in the incubator there also need to be water, because it needs to be certain humidity. I think I didn't note it on the long presentation. How the cells are proliferating. So we put them to the flask, and they attach to the flask, most of them, because some of them they don't attach. And then we need to provide a medium. Medium is like food for them, and this medium is supplied with different growth factor, hormones, proteins that make those cells proliferate, be alive, and be viable. So this is the time for your experiments. You take the cells and you cultivate them. You make your experiments. You make different manipulation, whatever you need. But for longer storage, because of course you cannot cultivate cells for all the time, right? So sometimes when you don't have experiments and you want to just store your cells, there need to be also some certain condition to store those cancer cells. So for short period they can be stored in minus 80 degrees. For longer period it should be like minus 140 or liquid nitrogen. And normally you prepare those cancer cells in the cryovials, in the small vials that can survive this very low temperature. And also you need to prepare a special solution that is here down. I just wrote what is that, so maybe when you need it in the future you already have the protocol how to prepare it. So normally you count number of cells. It should be minimum 1 million per one vial. And then you dilute them, you resuspend them with this solution, and then you put to your storage place. So now it comes to this, like a protocol part of this presentation. We will go through all those steps, because those steps are the most important in being able to cultivate cancer cells. First of all, it's the process of thawing cells. So when you have them in these vials that you have on the left side, 0.1 picture. Those are those cryovials, and thawing is also a very important process, because if you don't do it correctly they will die, because they are pretty sensitive for changing of conditions. Second thing is cultivation and passaging of cells, because of course when they arrive to being confluent in the flask, you need to passage them. You need to put them to the new place and give them new food, speaking very colloquial. So that's also not very complicated part. What is maybe a bit more complicated is counting cancer cells, because for most of the experiments you always need to know which number of cells you use. Because also sometimes you are plating cells in different small wells or something like that. So for every time you need to have exactly the same number of cells. So it's very important to be able to count cancer cells. And then the last step after you finish your experiment and everything, how to freeze down your cells and put them in the stock in the minus 140 or liquid nitrogen. So the first point, when you bring your cells from the storage place, so from the freezer actually, you need to bring them. The best would be to bring them in the dry ice, but at least in the normal ice, because the thawing process needs to be extremely fast, because they will die. You will lose a lot of cells if it will not be fast. So you need to transport them from the place where is your fridge to the place where you will put them to the culture very fast. And the thawing process itself occurs in the water bath. So normally you have water bath where it's 37. You put your vial without putting the lid also to the water, because then you can have a contamination. So you just put down part where is the cells, where is the solution, and then immediately when the cells become liquid, it's not anymore solid but liquid, you just pipette them to the flask with already pre-warmed and prepared medium for the cultivation. What is important also is that in the time when the cells will attach, because we will work with adherent cells, so they attach. In the moment when they attach, you need to change the medium, because the solution where you freeze them down contains the MSO, which is in high concentrations is very toxic for cells. So in the moment when they attach, you just remove the medium that you told them, and then you put the new one. So passaging cells, it's very easy. It's really easy. So you have those flasks that you see in the pictures. You open them under the laminar. You suck the medium. You wash them with pre-warmed PBS to remove the rest of the medium, which can interfere with the passaging. And then you remove the PBS, and afterwards you add trypsin. Trypsin enables us to detach the cells which are adherent to the bottom of the flask. So when you put the trypsin, you just break the peptide bind that bind the cells to the bottom of the flask. And after if you move a bit the flask, you can see if they are already detached or not. And then you just put certain number of medium, and you just mix it, and then you put whatever amount of volume to another flask and fill it with the new medium. So this step is a bit more complicated, and I would like to spend a bit more time on that, because that everybody understand how to count cancer cells. So first of all, you need to detach them. So the process for counting cells is at the beginning exactly the same like for passaging. You need to suck the medium, wash them with PPS, put the trypsin, and wait until they are detached. When they are detached, you can take 10 microliter of those cells into small tube, like Empedorf tube, and mix it with trypan blue. This is a step which is not 100% necessary, but I would say that it's important, because trypan blue enables us to understand which cells are alive, which are not. Because sometimes maybe in the process of passaging or whatever you are manipulating cells, they can die. And you of course don't want to count cells which are dead, because you want to count only cells which are alive for your further experiments. So that's why it's important to mix it with trypan blue, and then all the dead cells will be stained with blue. And then 10 microliter of this mixture you put to this hemocytometer that you can see on the picture, and you see that this blue thing here is already exactly this mixture of cells and trypan blue inside, ready to count. And then you go to the microscope and you count them. So this will be more or less, it's of course the picture only, but this will be more or less what you will see under the microscope when you will count your cells. The idea of this hemocytometer is it's important to count at least, because we have 9 different squares, and to be sure that we count a correct number of cells, you should count at least cells with 3 different squares, at least. But I always count from 5. I always count from the edges, 4 edges. I didn't want to do that. And from the middle one. But it depends. You can also, 3 is really minimum. So you count cells from 3 different, this is visible? Ah, it is visible, okay. From three different squares. And as you could see, like maybe I will show you on this picture, as you could see here, some cells, they will be attached like on the edge of the squares. Because you know, one square is, it would be better if I would have a laser. So one square is this one that I mark in red. And some cells, as this one here, they are just attached to the edge of 2 different squares. So to be sure that you don't count the same cell 2 times, you always need to say to yourself in your head, it doesn't matter which one, but you need to know by yourself, that you will count cells only from either left, like here they are marked with black and white. Either you count cells which are attached to the left upper part of the square, or right down. You understand why? You understand why, or you don't? Because you understand, okay. Because you know, like after you can count the same cell, because you know, you count everything what is on the line, and then you count one square and the second, and you count the same cell second time. Of course, then the number is not correct at the end. And these hemocytometers are designed like that the volume that you put in one of those squares, so this one that we see, it's minus 10 to 40 milliliter. So this is the volume. It's important for after the calculation. I mean, if you want, I didn't put the formula how they calculated it. You can Google it if you want, because I thought it would be too complicated, too many numbers and everything. But this is just like something that you always know. This is always minus 10 to 40 milliliter in every hemocytometer. It's just standard. So now the counting cancer cells. As I said, you need to count first from the three squares, always, and make the average of that. So here in our example, the average of counted cells is X. First of all, what you will calculate is called cell density. So you will calculate how many cells you have per 1 milliliter. So the formula is that you will multiply the number of cells, your average of course, multiplying with the dilution factor. Dilution factor, what does it mean? As you remember when I said to you how we prepare cells, we are mixing the cells together with trypan blue. So when we have this mixture, it's diluted, right? It's not exactly what we took from our flask. Since we diluted it with trypan blue, we need to, after in the calculation, take it into consideration because we diluted 2 times. So that's why our dilution factor is 2. And then we divide it by the volume that is in 1 square of this hemocitometer. And afterwards, the cell density that we will have, it will be number of cells that we have in 1 milliliter. But of course in our flask we had, let's say, 10 milliliter of cells. So to be able at the end to get a total number of cells, you need to multiply it by the total volume of medium that you had in your flask. You understand? I think it's very complicated if you don't see this, I know. But I decided to prepare also some kind of example because maybe with numbers it will be a bit easier to understand. So, as you can see on the square, we, let's say, calculate from the 3 squares A, B, C. So in first square A, we count that there is 11 cells. In the second square B, we count that there is 13 cells. In the C, we count that there is 9. So the average is, of course, we sum everything and we divide by 3 because we calculated in the 3 different squares. And we count that we have 11 cells. So the next step is calculating the cell's density. So we multiply it by the dilution factor, which is 2 because we diluted with the trypan blue 2 times. And we also multiply it by 10 to 4, because it's easier to multiply it to 10 to 4 than to divide by minus 10 to, it's just mathematics, it's just the same. And then we get at the end a number which says to us how many cells we have in 1 milliliter. And after, to be able to get the total number of cells that we have in our flask, we just multiply it by the number of, by the volume that we have in the flask. And it gives us the total number of cells that we have in our flask. And then, you know, when you know how much cells you have, after you know how much cells you need for your experiment, so at the end you know how big volume you need to take to be able to obtain this number of cells for your experiment. So did you get this calculation? Because I think this is the most crucial point of. If you have some question, please ask me. I can say it again. So now I prepared a test for you. So we have 3 squares, and then please, if everybody can calculate how many cells we would have in this condition that you don't need calculator. I design it like that you can do it with your head.
You want me to do it here?
Yeah.
Ah.
And if you don't want, then it's okay.
It's 400.
You got the number? Catherine, how much you got? Very good. So it means that you understood. I'm very happy. Because I was really afraid that it would be a bit complicated, because I think it's easier to do everything and to understand everything when you see it and when you do it. You got?
Yeah.
Or no? You got?
Yeah.
Okay. Francis?
Yes.
Okay, good. So afterwards, from the previous example, it was 330 cells times 10 to 4, and the volume of our flask was 15 milliliter. And let's say we say that we need for our experiment 1 million of cells. That's just example, of course, this one. So how we calculate, you know, is this basic thing in the mathematics that you make this cross calculation. You know, if you have 330 cells, 330 times 10 to 4 cells in 15 milliliter, in how many milliliter you have 1 million of cells. You understand that, right? It's just a cross calculation. This is very easy. And then if you calculate it, it's 4.4 milliliter. So it means that from your flask you need to take 4.4 milliliter of your cells in the solution, in the medium, for your further experiments. Because then this amount of volume contain 1 million of cells. So then the last point is how to freeze down the cells. So I already mentioned in the condition what is the protocol to prepare the freezing solution. It's 20% of FCS and 80% of DMSO. And this solution you mix 1:1 in dilution with a medium. This will be already ready for putting your cells in the vials. And then of course you need to count your cells. Because in one vial it should be at least 1 million of cells. And then when you count that you have 1 million, you just spin them down, you make a pellet out of cells, and then you put 1 milliliter of your, did I write this one? No, I didn't write that, I wrote. 1 milliliter of the freezing solution, and then you just close the vial, and then put it to your storage place. And that's it. Normally people say that freezing them down should be a bit slower process, in contrary to the thawing. So you shouldn't put them right away to minus 140. You should put them first to minus 80 and then put them to the... But from my experience, I know that it doesn't harm cells. I mean, I did it many times and it doesn't. But maybe it depends on the cells. Maybe I was working with very strong cancer cells which are not very sensitive. But this is what people say that you should... So that's it. Thank you for your attention, and since it's my last day today at work here in Chiromo, I also wanted to thank all of you for collaboration for last months that we spent together, and we did some things together. And if you have any question, you can ask me now or later when we will do the practical part.
First of all, we clap for you.
Thank you.
So that we also give you a very good setup, right? So, there you go.
Thanks.
Me, actually, without asking a lot of questions, first of all, I would say I appreciate your effort for coming up with this kind of presentation. Hopefully, you're going to put it on our emails as usual. And I believe also that whatever skills you have, you're going to impart to us, you're going to speak. Okay? And as you go, I think you're going to leave a team of people who are actually very well trained, who will proceed with a set of study. Even where you are, you'll be hearing that you're publishing. Your name will always appear on those papers as we move on. So actually, we are very happy. Now, the other thing I wanted to clarify is that, as you are saying, because I remember I was working in the cell culture back in 1986, those are the days. And one of the things I was trained is that, as much as you are saying that the cells don't get hurt when you are preserving them, when you're putting them at liquid nitrogen, that is minus 196, 140. I was actually told that it should actually be gradual. You actually move from minus 20.
No, nowadays they said that you should put them first in minus 80.
Minus 80, and then you move on to the liquid.
Exactly. This is what they said.
Especially, and more so, if you know there is also this other preservative, the glycerol. Glycerol, probably you can use the DMSO or use glycerol.
Yeah, actually I never worked with glycerol. That's why I didn't put it, because I wanted to say to you how, It doesn't mean that everything what I'm saying, it's the only one possibility to do it. Yes. It's just what I knew how to do it, and I didn't want to make it, because then it would be too complicated. You know, I also didn't mention, for example, that there are two ways of passaging cells and cultivating cells, because some of cells they are adherent, some cells are floating, so it's a bit different way of handling. But since now we have cells which are adherent, I just prepare it for that, just to not make it too long and too complicated, because after it will be just,
And in this particular case, whatever the cells we are using right now, they actually form monolayers on the surface of the culture cells.
This that we have,
That's what we have.
Actually, I didn't even manage to check which kind of cancer are those cells, because because you know, I just rushed here, because I didn't know which one because at the beginning we were supposed to have prostate cancer, but since the incubator didn't work, I just asked them to split for me whatever cells they have which are cancer. So they split for me, but I don't know those cells.
Know which ones
we need to check them to Google which kind of cancer it is.